In this section of the class, we're going to start thinking about the chemistry behind how different organisms can control whether they are creating energy through glycolysis or storing that energy in different forms. Okay? And we're going to think about this in two perspectives. One is we're going to think about some signals inside the cell that communicate a high energy status or a low energy status. And the other way we're going to think about it is how the conditions in the blood can influence the metabolic state of cells. Okay? And the latter really boils down to two really important hormones. One is glucagon and the other is insulin. So both of these hormones are secreted by the pancreas. Glucagon is secreted in response to low concentrations of glucose in the blood, while insulin is secreted in response to high concentrations of glucose in the blood. Okay, so glucagon and insulin flow through the blood, and then they can interact with different cells. All right, so glucagon interacts with the cells through a G protein coupled receptor, whereas insulin will interact with those cells through a receptor tyrosine kinase. Okay, but they both can communicate information inside the cell. Right? So glucagon's primary target that we're going to consider is the liver. So when glucagon sends a signal inside the liver, it's telling the liver that there's a low amount of sugar in the blood. So what the liver does is it reads that signal and recognizes it needs to break down stored sugar or glycogen and then release that sugar back into the blood. Okay, that way we can start to rebalance the amount of sugar in the blood so that your brain doesn't starve. All right, so the opposite's true when insulin interacts with the liver. It sends a signal that ultimately tells the liver that we need to use up this glucose in the blood. So it will uptake the glucose and store it as glycogen. Insulin will also interact with other cells. For example, adipose tissue. The signal that insulin sends to adipose tissue would say, I have a lot of energy, so I need to store it. And adipose tissue will store that energy as fat. Okay, so in the rest of this introductory video, what we're going to think about is just some basic ideas behind how this control can happen. And then over the next several videos, we'll explore this in more chemical detail. We'll start off by looking at a simplified scheme of glycolysis. So what we're looking at here is the conversion of glucose to pyruvate and eventually to energy. Note that I've only highlighted some of the steps, and in this case, really only the first few steps. And remember that two of these steps are considered irreversible because of the high energy demand it would take to convert glucose 6-phosphate back to glucose or fructose 1,6-bisphosphate back to fructose 6-phosphate. Okay? But as it turns out, those steps actually are reversible. They just use a different enzyme and catalyze a little bit different reaction. And what this means is that we really are able to reverse glycolysis. And when glycolysis is reversed, we call it gluconeogenesis. We mentioned earlier that glycogen is able to be synthesized and that will store glucose. The synthesis of glycogen happens as a branch off of glycolysis, and it starts at glucose 6-phosphate. And depending on the energy status of the cell and the sugar status of the blood, we may want glycogen synthesis turned on, but we also may want it turned off. So there has to be some way of controlling whether we're making glycogen or not making it. And that is really the big point of the rest of the videos in this unit, is to think about how we can control flow of metabolites through glycolysis and also control whether we are synthesizing glycogen or breaking glycogen down.